Good afternoon, cultures. So, my name, before we get started, I'd like to introduce myself. My name is Freezy-Bee, and I'm with my colleague. So, today we are going to teach you about life science. We are the new teachers of life science. So, before we get started, what do you understand about life science? Okay, what is life according to you? Yes?
All you can find inside the world.
So life includes all living organisms. Living organisms can be plants, animals, and humans. So now, what is science? What do you understand about the word science? Okay, science, it is the process of finding information or facts regarding nature. It can be made through a hypothesis, observation, and some other external methods. So today we are going to focus mainly about DNA, RNA, and protein synthesis. So without wasting any time, my colleague will take over and teach you about DNA. You can take over this.
How are you guys? So good. So we're going to be looking at the two types of nucleic acids, DNA and RNA. What do you know about DNA?
DNA is called life.
Yes, DNA is the code of life. Yes, come on. Right. So we are going to look at the structure of DNA. Can all of you please do this with your fingers? Imagine it as a long string of that shape. That is the shape of DNA. Yes. So we're going to be looking at the differences between DNA and RNA. DNA consists of deoxyribose sugar. You just take the part where, okay, does anybody know the meaning of DNA? Like the full name of DNA? Yes, Kato.
Deoxyribonucleic acid.
Yes. RNA? Anyone? Yes. So I want you to take that first part there. It's deoxyribose and ribose. Those are the different sugars between DNA and RNA. So in DNA we have deoxyribose sugar, and RNA we have ribonucleic ribose sugar. Yes. And then the shape DNA is double helix. What about RNA?
It is stranded.
Yes. And then we have nitrogenous bases. How many nitrogenous bases do we have? 5. Yes, that is true. So, one type of nucleic acid consists of 4 nitrogenous bases. In DNA, we have thymine instead of uracil. So, adenine because of thymine, guanine with cytosine. What about in RNA? What does adenine pair with?
Uracil.
Yes. So DNA is found only in the nucleus. Then RNA is found in nucleus, ribosome, and cytoplasm. Yes. So now we're going to be looking at DNA replication. So DNA replication, it is the process where DNA makes an exact copy of itself, right? Yeah. Where does it occur? Interface. Yeah. So now we're going to be looking at the process of DNA replication. How DNA replicates itself. Yeah? So we're gonna, okay. Can you please do this again? The first step is the DNA double helix unwind. Can you please unwind? Yes. breaks, ne? So the weak hydrogen bonds separate the nitrogenous bases, ne? And then you're gonna form two DNA strands. It's gonna be one strand and one strand, ne? And then the free-floating nucleotides, they will complement each other on the free DNA strands, ne? So adenine is going to pair with guanine is going to pair with yes. Okay, so now I'm just going to go through the process, ne? So firstly, what does it do? Yeah, and then the second step. Yes, and then each original DNA strand serves as a template. Yeah. Then for number 4, free nucleotides build a DNA strand onto each of the original 2 strands. Yeah. Then this is going to result in 2 identical DNA molecules. Each strand is going to consist of an original DNA strand and a new original strand. Right? What is it going to form? DNA what? Okay, so now we're going to look at the significance of DNA replication. Why does it occur? It doubles the genetic material so it can be shared between the resulting daughter cells during cell division. And then it's going to result in the formation of identical daughter cells during mitosis. I'm gonna give you a live practical of DNA replication. Maybe you're busy in the kitchen, maybe chopping some onions, and then you cut yourself. Take your finger as a DNA double helix strand. It's gonna do it. It's gonna unwind and unzip the weak hydrogen bonds that are supposed to stick together your skin. They're gonna break, right? Yes. Then that's when DNA replication takes place. Then to form the new skin, then to just attach the separated skin, right? Now we're going to look at DNA profiling. Okay, so it is a scientific procedure that is taken in individuals to look at their bands, yeah. So why does it occur? Yes, why do we do DNA profiling?
To identify criminal.
Yes.
To find long lost family members.
Yes, that is true. Yeah. That's all you should know about DNA profiling. Any questions?
No.
No questions? Okay.
Thank you, Ms. Quay. So, I'll be teaching you about protein synthesis. What do you know about proteins? Like what is protein? When do we find a protein? Yes? We find protein in the meat, in the milk, eggs, right? So proteins are very essential for the body. It builds the tissues. The body contains thousands of tissues. So what is protein synthesis? A protein synthesis is a process by which proteins are made in each cell, forming enzymes, right? So in protein synthesis you should know, I think 6 or 7, if I'm not mistaken, the terminologies that are very essential for this topic. You should know amino acid, base template, transcription and translation, and colons, and also anticolons. We'll start with amino acid. When you hear the word acid, what do you think of?
We go danger.
Danger, right? Because the word acid is strong by itself. But in this situation, they are not referring to danger, but they are not referring to danger. Amino acids are the building blocks, or we can say monomers of proteins. What are building blocks? Building blocks, it means that for proteins to occur, you need amino acids, right? Am I audible enough at the back?
Yes, yes.
So, and then secondly, you should know the base template. What are the base template? Yes, when I talked about them. What are those?
Each triplet or
I'll give it, we'll start with A and pair with T.
or you adenine, thymine, guanine, cytosine.
Yes, those are the base templates. The nitrogenous base of So now we focus on transcription. This transcription, it is the first stage of protein synthesis. It is where the proteins are made. It starts in transcription and followed by translation. So the transcription is the first stage of protein synthesis. It is where the mRNA from the DNA carrying code for the protein to be made. It means that when transcription, it occurs, you should know what it occurs. It occurs where? In cytoplasm. Right? Yes. It is where transcription occurs. For transcription, you should know this terminology. The nitrogenous base on DNA are called nucleotide. Yes. But on our transcription, they are called codons. Right?
Yeah.
So, what are the codons? Codons are the building blocks of the mRNA. mRNA stands for messenger ribonucleic acid, right? So, it is the messenger. It carries the proteins that will pair with the other amino acids that are coming from the translation, which will go there after the transcription, right? So, transcription, it is essential because it happens where it takes its complements from the DNA replication, which occurs on the cytoplasm. So after DNA replication, our next step is transcription, where our mRNA will be formed with the new strand that was formed from DNA replication, right? So DNA replication gives us one strand. That strand will serve as a template for another strand that will be mRNA. Right? So after mRNA, it will leave through the nuclear pore on the nucleus, right? It will leave. It will travel to the ribosome. Right? Remember, it is the process of forming protein, right? So the second stage is translation. Do you understand what I just said, right? Yes. So translation is the second stage of protein synthesis, which carries the translation, carries the amino acids, which combine to form a protein. You know what I mean? So what are the amino acids? I said amino acids are what?
The monomers of the protein.
Thank you. Monomers of the protein. Those are the amino acids. So the amino acids are something like it has shapes. So the RNA is the transfer ribonucleic acid. It transfers the amino acid to where? To the ribosome. When it attaches to the ribosome, and then it will pair with the family that are attached to the ribosome, right? And those, at the top of the RNA, we have what we call anticodons. Anticodons are the monomers of the RNA, right? So remember, we say DNA, it has what? As a nitrogenous base, what we call it?
Nucleotide.
Thank you, nucleotide. And then the mRNA, the codons. And then RNA, anticodon. Right? In simple terms, we say it will be something like A, this A has what? G, right? G has A and G. nucleotides. And the second is what we call the codons. And in here, we have anticodons. Can someone tell me what you spot the difference here?
The change. The change.
The difference is there. Right? So as you understand now, and we did say protein, it is the process by which proteins are made, right? So now we are going to focus at the peptide. Here the peptide, as we said, you see how do they look like? Those are peptide. These genes found in DNA contain the code which determines which protein will be formed, right? So the smallest protein contains about 50 amino acids linked together. So it means this thing is very long. It contains about 50 amino acids, right? So, I think I've summarized everything. So right now, I'm ready to take any questions if you understand. Yes.
The effect of mutation on protein starch.
The effect of what?
Of mutation on protein starch.
The effect, the change, the mutation is the change in the nucleotide. If we call mutation, the mutation will cause what we call a transfer, which will cover that topic on genetics. Now let's start. Are we clear?
Yes.
Okay, so right now we are going to take a quick quiz to show what we understand. So we'll be asking you some easy questions about what we just covered, right?
Yes.
Okay, what does DNA stand for? Okay, where in the cell is DNA found? Yes.
Nucleus.
Which pairs with adenine?
Which has
Which pairs with adenine?
something like that.
Thank you. So for today, I think we are done. Okay, for your homework, please, like find the DNA extraction. What is the DNA extraction? And where do we find?
Yes.